Yeah, by the way, if you guys have any questions, you can stop me and ask anytime. Okay, feel free. So I'm going to start with these slides. So on the left side, each picture is, each of these pictures represents one of these medical conditions listed on the right side, to include Parkinson, cortisol, mouse ulcer, leukomalacia, Kaposi sarcoma, and oral warts. I think you guys probably have already learned a little bit about this from Tim's class. Is that right? So maybe you can recall. Can you guys match up? Tell me what is what? Which is which? Yes.
I think A is leukoplakia.
A?
Yeah. Oh, no.
Okay, so it's okay. So I can tell. It's okay for you guys now to be able to tell right now, and hopefully, by the end of my talk, my lectures, you will be able to know exactly, everything about this. What I mean by that is that you will be able to not only learn the disease, each of these, but also the pathogens that are associated with each of these conditions. Okay. And also, I want to tell you that most of these conditions are associated with the DNA virus infection. Okay. So here's the study objectives. Okay. So as a dentist, I think you will be the first one to tell your patient what they have. So in order for you to tell your patients what they have, you first of all need to learn each of these different medical conditions, infections, and the disease, and also the pathogens, and understand how the disease, and the pathogens transmitted, right? And also how the disease occurs, and also how the disease progress, develop, and ultimately you should be able to tell, be able to distinguish the different medical conditions and make a right diagnosis. And so you can refer your patient to the right doctors for treatment, right? So this is why you are here, basically, for these lectures. So here's what my plan. So first of all, I will give you a brief introduction about DNA viruses, and then I will give you more specific details on each of those different type of virus, DNA virus, what they do, what they cause, what kind of disease they cause, and how we treat them, how we diagnose them, right? So first of all, I'll give you an introduction. So DNA virus is basically a classification of viruses based on their genome. So based on the genome, there are two types of viruses: RNA viruses and DNA viruses. So the RNA viruses, obviously their genomes are composed of RNA molecules, right? So DNA viruses, their genome are composed of DNA molecules. And there are many different DNA viruses, many. So we can further clarify, clarify this DNA virus into three groups. The first group is those viruses that contain genome that is a single-strand DNA. And the second group is those viruses that have genome that is a double-strand DNA. And also the third group is viruses their genome is partly DNA and partly RNA. Okay, so our universe is, is really diverse. There are all kinds of organisms. So here is another way to classify DNA viruses based on the various structures. As you know, some viruses, they have membrane, or what's called envelope outside of the viral particles. So this is called the envelope viruses. And then also the other group of viruses, they don't have an envelope the outside of the viral particles. It's just a free viral particles. They don't have an envelope. Okay, the envelope comes from the cellule, from the cells from which the viruses are produced. Okay, when the viruses are produced from the cells and get out of the cell, they will acquire the membrane. Okay, which I will talk about later on. So among the envelope DNA viruses, and there are three groups. Well, the first group is the parvovirus. Okay, these are the largest DNA viruses that has that caused millions and millions of people deaths in Europe in the 18th centuries. Basically, this virus is associated with so-called smallpox. This is a very deadly disease. Killed so many people in Europe in the 18th century. And because of the development of vaccine by a British doctor, so people were able to fight this disease. So the vaccine has been very effective and widely used in the whole world. So as a result, the World Health Organization declared eradication of this virus worldwide in the 1970s. So now, right now, we don't see this virus anymore. We don't see the disease anymore. Okay, so that's why I will not cover this virus in this lecture, because just let you know that this, you're not going to see this anymore in your career, in future career. You're not going to see this. Okay, but it's a DNA virus. It's the largest DNA virus. It's very big. The second family of this envelope DNA virus is herpes virus, which I will spend a lot of time to talk about. And the third group of this envelope virus is the hepatitis B virus. I will not cover this. I think there will be another lecture on RNA virus after me. So I think it will be covered by that lecture on hepatitis. So the hepatitis B virus is very unique. Its genome is part of the DNA and part of the RNA. Okay. So this is the envelope viruses. And then among the non-envelope DNA viruses, was further divided into two groups. One is double-stranded DNA viruses, and the other one is single-stranded DNA viruses. So among the double-stranded DNA virus, one group is adenovirus, which is a small virus that causes flu symptom like flu-like symptoms, disease, and people often get infected by this virus, and you think it's a flu, but it's not. It's actually caused by this virus, adenovirus. But it's not something that is serious. Usually it's a self-recovery disease. Without treatment, the virus will be cleared about 2 weeks after infection, and you will recover. So it's not something very serious. So I will not cover this virus in this lecture. You guys just need to be aware that it's a DNA virus. Okay. The second group of this envelope, small DNA virus is polyoma virus and also papilloma virus. So these are very deadly viruses, and they can cause cancer. And I will spend quite a bit of time to talk about these two viruses. So this single-strand DNA virus is the so-called power virus, which is the smallest DNA virus, and its genome is a single-strand DNA. And this virus usually cause skin rash, in little children. It's something not very severe. It's after a few days, the skin rash will just go away without treatment. In immune competent, immune surprised kids, if the kids have immune suppression, it can cause anemia. But other than that, it's not something serious. It's not something very important, and that's why I will not spend too much time on this virus either. So this table just summarizes what I told you. On each of these type of DNA viruses, you guys can spend some time to read through this slide. To have an idea of each of these viruses. Okay, compared to RNA virus, here are the common features of different DNA virus. So you may learn later on from the other lecture on RNA virus. Different in the RNA virus, DNA virus are replicated in the nucleus. Okay. RNA virus, you may know, most RNA viruses are, for example, influenza virus, they replicate, in the cytoplasm. Once the virus get into the cells, right? So DNA virus is different. Most DNA virus replicate in the nucleus, not in the cytoplasm. And the other difference between RNA and DNA virus is, RNA virus, they mutate so frequently. So, for example, I'll give you an example, HIV virus. Okay, when you get infected by HIV virus, the virus mutates so quickly, and that's why we have such a hard time to develop a vaccine against HIV virus, right? Because it mutates, constantly mutates. So even your body develop an antibody to fight the HIV, but the virus mutates and then generates a mutant, so your antibody does not fight the initial virus you get. The same thing with influenza virus. That's why, because the influenza virus mutates so quickly, and every year you need to have a different shot, right? Compared to RNA virus, DNA virus is not like that. They are relatively stable and they don't undergo very frequent DNA mutations. Okay, so that's the difference between RNA virus and DNA virus. And the other thing is about DNA virus. Most DNA viruses, they are oncogenic, which means they can transform normal cell into a malignant tumor cell, because they have the ability to suppress tumor suppressor genes and also activate oncogenes once they infect a normal cell. So that's why they can make normal cells become a tumor cell. Other viruses usually don't have that ability. And the only exception is leukemia, virus that can transform a normal cell to become into a tumor cell. The other difference between RNA viruses and DNA viruses is that RNA virus, when you get infected, within a couple of days or a couple of weeks, your body will be able to, your immune system will be able to fight the virus, clear the virus away, and so you can recover completely and you will become virus free right after you recover. So DNA virus is different because most of DNA virus, once you get infected, you are going to have a lifelong persistent infection. In other words, the virus will stay in your body for the rest of your life. Okay, and this is a very, very unique. The other virus, except HIV, most, for other virus, in order for them to have a persistent infection, they have to integrate into your host genome, for example, HIV. So once your cells are infected by HIV virus, the virus genome integrated into your host cell. And then, so that's why you are not able to clear the virus, and the virus stay in your body, right? But most of the other virus will be cleared after recover by your immune system. But dengue virus, is unique. Most of the time, most of dengue virus will stay in your body and have a lifelong consistent infection. Any questions so far? Yes.
For HIV, if it's constantly mutating,
Yes.
do we ever see random loss of function mutations?
That may actually happen, but we really don't know. I think the virus mutates is an advantage for the virus to stay in our body. It's a way to evade the immune system, immune surveillance. Okay, and that's why it's so hard to get rid of the HIV virus. Because it's constantly changed, constantly making mutants. Okay, it's a way for the virus to avoid, to evade your host immune response. Now I'm going to get into each of the type of the dengue virus. So the first group is the human herpes virus. Of course, herpes as a virus is a big family. I mean, it's not just affect human, but also affect the other species. All animals they have their own herpes family version. But in human, we have human-specific herpes virus that can only infect human. So here is a particular, in the typical structure of the herpes virus. Okay, so inside the virus particles is the double-stranded DNA genome. Okay, it's a very large DNA genome, double-stranded linear DNA virus, linear DNA molecule. And the virus particles basically is this kind of form. It's called icosahedral form. It's basically made of the virus protein, capsid protein. Okay. So the DNA is packaged into these capsid particles. Okay. And then on the outside of these virus particles is the membrane or the amidon that is required from the cells. And in between these virus particles and the membrane is something called the tegment. Basically, the tegment is a whole bunch of different virus proteins that serve as a matrix to further protect the virus particles. Okay, it's just like a matrix. Okay, to protect the virus particles. So as I mentioned, this is the amidon virus, right? So the membrane come from the cells when the virus is produced by the cells. And so far, we have discovered 8 different herpes viruses in human, called herpes human herpes virus 1 to 8. So based on what type of cells they infect, we can classify these 8 different herpes viruses into 3 groups. Yeah, the first group is called alpha herpes viruses, which include the herpes simplex 1, herpes simplex 2, or HIV1, HIV1 or HIV2, means human herpes virus 1, human herpes virus 2. Okay. And also varicella zoster virus, or basically is what's called human herpes virus 3. Okay. So basically, this alpha group of herpes virus, because they can infect neurons in the brain, so they are neurotropic. Okay, which I will talk about in more detail later on. And the gamma herpes virus basically include Epstein-Barr virus, or EBV, what's called human herpes virus 4, and also Kaposi sarcoma herpes virus, or HHV8. So these gamma herpes virus, they can infect lymphocytes. They are lymphotropic. Okay. And this The other group is beta herpes virus, basically include the cytomegalovirus or CMV, and human herpes 6, and human herpes 7. Okay, so this group of herpes virus, they can infect a variety of different type of cells. They have have broader range of tropism. So that's why they are grouped as beta herpes virus. So as I mentioned, all herpes virus, they have a huge genome, running from 120 kilopair pieces to like 350 kilopair pieces. They can encode like more than 100 different genes or proteins, so they are huge. Inside the virus particles, the virus genome is a double-stranded linear DNA. Okay, so linear DNA molecules, and at each end of the linear DNA molecules, you can see there's something called direct repeats. Okay, basically these repeats are tandem repeats, and there must be copies of the same DNA sequence. Okay, like 100 copies. So
What were the DRs?
it's called direct repeats.
And what do they do?
I'll tell you a little bit later. Okay, these are basically composed of the same DNA sequence, tandem repeats. Okay, so this repeats serve multiple purposes. First of all, when the molecules is in a linear form, these tandem repeats will serve as a way to stabilize the DNA molecules. Okay, to protect the DNA molecules. And also as the virus get into the cells, okay, when the virus get into the cells, eventually the virus will eject the DNA or the genetic materials into nucleus. So once the virus DNA is in the nucleus, it will become a circular DNA molecules. And this has a lot to do with this direct repeats. As I mentioned, these direct repeats are composed of the same sequence. Okay, they are homologous, 100% homologous. So by homologous recombination, okay, between these two internal repeats, these linear DNA molecules become circularized. Okay, and that's why that's how it occurs. Okay. So once the viral DNA is in the nucleus, and we'll make multiple copies. As as I mentioned, we have 8 different herpes viruses, and each virus infects a different type of cells. So what is that? So the reason for the different type of virus to infect different type of cells is because the virus needs or requires a receptor for the cells or to get into the cell, right? So it's like HIV. You guys probably already know that in order for HIV virus to infect CD4 T cells, HIV targets specific target immune cells, CD4 T cells, right? That's because CD4 T cells express specific receptor for the HIV virus, and which is CCR4, CCR5 protein, right? And the same is true for herpes virus. Each of these herpes virus, they also needs a receptor for the virus to recognize the cells, the target cell. Okay, so basically, I just give you some examples for each of these herpes virus. For example, for herpes simplex 1 and 2, they can use TNF-alpha receptor and also immunoglobulin-like proteins as the receptors for the virus to get into the cell, to recognize the target cell and then get into the cell, right? The virus, roster virus, for example, they use mannose for 6-phosphatase receptor to get into the cells, right? And also the other, these are the receptors for the other herpes viruses, okay? You guys can take a look. Basically, what I'm telling you is, each type of these herpes virus requires a specific receptor to get into the cells. And that's why the herpes virus, each herpes virus infects a specific type of cells. So this table just summarizes, each type of these herpes virus, what type of cell they infect and what type of cells, for example, the alpha group herpes virus, herpes simplex 1, when they first infect a cells, they infect the mucosa epithelial cell, right? Later on, they also can infect neurons, and they are neuron trophic. The same thing is true with herpes simplex 2. When they first infect you and they target the mucosa epithelial cells, later on they can get into neurons as well. The lymphotropic herpes virus, for example, cancer disease or hepatitis 8, they first infect B cells. Right now we don't know when when they get into the infection, we don't know which cell they target, but in general they target B cells and T cells. Okay, so basically each type of these herpes virus they target specific cells. Okay. Now here's what happens when a virus attacking during. This is how the virus infects the cells. Okay. As I mentioned, each virus requires a receptor, right? So when the virus recognizes the cell receptor, then they will bind to the cell surface, right? Once the virus is bound to the cells, okay, it can get into the cells through two ways. One is through endocytosis, okay. So through endocytosis, the viral particles can get into the cells. And the other way is because the herpes virus has a membrane, right? And the membrane comes from the cells. So the membrane of the virus can fuse with the cellular membrane, okay? So through this fusion process, the virus can get into the cells as well, right? So through two ways, the virus can get into the cell. And once the virus is in the cells, it will travel through the cytoplasm and then get into the nucleus. So because the virus, herpes viruses are so big, once they get into the nucleus, they will be blocked at the nuclear pore. Okay, the nuclear pore is very small. The whole virus particles are too big, so they cannot cross this pore and get into the nucleus as a whole virus. Instead, what they do is the virus will eject its DNA into the nucleus. Okay, and as I mentioned, once the DNA is in the nucleus, the DNA molecule will become circular. Okay. Any question on this process? Okay. So once the DNA is in the nucleus, okay, it will start to express the various genes, right? So in the start, the first gene that expressed, I'm talking about the various genes. The first various genes that are expressed are called something called immediate early genes, or IE genes. So these are the first genes that are expressed once the virus DNA is in the nucleus. So usually these immediate early genes, they are virus encoded transcription factors. Basically, can serve as a transcription factor that can turn on expression of other virus genes. Okay, so and then it follows by the expression of the early genes or E genes. So these early genes basically encodes proteins that are involved in virus DNA replication. Okay, and then it followed by the expression of mid genes. The mid genes are basically encodes various structural proteins like the capsid, the integument, etc. So once the virus DNA, and then it followed with DNA replication, of course. Basically, the viral DNA will make multiple copies. Okay, so once the viral proteins are made and the viral DNA are made copies, they will form new viruses. Okay, once the new viruses are formed, they will get out of the cells through exocytosis. During this time, they will acquire the cell membrane. Okay, so that's why the virus have an envelope. So it's important to note that during this process of this infection, when the virus replicates, okay, that's when the disease occurs, because when the virus replicates in the infected cells, okay, it will eventually kill the cells. Not only will kill cells, but also it will induce your host immune response. And that's when your cells, your body will produce a lot of inflammatory cytokines to counterattack the virus infection. Eventually, it will clear the virus and get rid of the infected cells. So that's why you will have a lesion during this infection, primary or acute infection process. And the other consequence of this infection is during this process, you will produce a lot of other viruses, new viruses, that can cause further new infections. Okay. So this is what happened to the cellular level, okay, during the virus infection process. But here is what happened systemically during the herpes virus infection. Okay, that's what happened to your body. So once the virus infect you at the mucosa, for example, when you first get infected, this I just talked about. So basically, the virus will replicate and make a new virus, and then the newly generated new virus will infect the other cells. Okay, eventually, in some, during this process, as far as you are immune competent, your immune system will clear this virus, so you will become virus free. But in some cells, the virus will get into a so-called latent stage. So during this latent stage, the virus genome will become silent, and there's no virus gene expressed. Okay, this is a way for the virus to escape your host immune response. Okay, so basically, the virus genome does not express any virus protein during this process. Okay, minimum level expression become dormant basically. So this is something we call the latency. Okay. From time to time, this latent virus can still reactivate. Okay, and so when the latent virus reactivate, we are making new viruses. Okay, and the new virus will transmit it and infect other people or infect other cells in your body, and to cause recurrence of the disease. Okay. So this is why once you are getting infected by a herpes virus, you will never get rid of the virus, because the virus will stay somewhere as a latent stage. It's very difficult for your immune system to get rid of it. And basically, once you have it, you are going to have it for the rest of your life. I just want to mention that everybody gets at least one type of herpes virus infection, including all of you. Each of you is infected, at least by one herpes virus, for sure. So this table I already showed you before. So basically, just again to tell you what happens for each type of herpes virus. What happens during the acute infection. So what type of cell they attack during acute infection. For example, herpes simplex 1, herpes simplex 1 or 2, they attack the mucosa during acute infection. And later on, they will travel to your brain to infect the neurons. where they stimulate the infection, where they have a latency in your brain. Okay. For the herpes 8 virus, for example, the first one, when they get infected, you get the target B cells, and then they will be latency somewhere. Okay, we still don't know. Anyway, so this table you guys need to read it through and just be aware of, what type of cell, the first attack and which cell the detection latency. Okay. And all herpes virus they transmit through contact. Okay. That's why everybody gets herpes virus, because they transmit through contact. As far as you get contact, you are going to get the virus. So as I mentioned, the latent virus can reactivate, and usually immunosuppression is so the latent virus reactive when most time it happens when you are immune suppressed. For example, if you take a medication for immunosuppression, or you have something happened to your immune system. If you are infected by HIV, for example, you have an immunosuppression, and in this case, you are going to have the reactivation of herpes virus. But the immunosuppression basically provides an environment for the herpes virus reactivation. Okay, because your body has no way to clear the virus. But immunosuppression is really necessary for the virus to reactive, but it's not sufficient. You need something to stimulate the virus to reactive. Here are basically the causes, possible stimuli that can stimulate the latent virus to reactivate. One thing is stress. So if you are stressed, first of all, when you are stressed, your immune system is going to be going down. Secondly, stress itself can trigger the reactivation of the latent herpes virus reactivation. The other way for the latent virus to reactivate is when you are infected by another pathogen. For example, I just mentioned the HIV, but also other pathogens. For example, if you are infected by bacteria or fungus, sometimes it can also trigger reactivation of the latent herpes virus. And most of the time, when you are infected by something, you have inflammation, right? So when you have inflammation, your body produces a lot of inflammatory cytokines. And this inflammatory cytokines can also trigger reactivity of latent herpes virus. Other causes, for example, when you eat too much meat or warrior, when your body produces too much reactive oxygen species, in which with something we call you undergoing oxidative stress. Basically, your body produces more higher level of reactive oxygen species than oxidant, right? So in this case, it can also trigger reactive virus. Another thing that can trigger latent virus reactivity is metabolic byproducts, such as short-chain fatty acids produced by some type of bacteria. Related to you guys, a lot of patients, they can develop periodontal disease. When you have periodontal disease, you are infected basically by a group of bacteria, right? Those anaerobic gram-negative bacteria that cause periodontal disease can produce this kind of metabolic byproduct that can cause reactivity of latent herpes viruses. Of course, there are many other possible stimuli as well. So basically, in order for the latent herpes virus to reactivate, first of all, you need to have an immunosuppression, which provides the condition, the environment for the virus to reactivate. But on the other hand, you also require this kind of stimuli for the virus to reactivate. Any question? If you guys have any question, you can just stop me and ask. Okay, feel free.
Ten forty-three.
So you guys want to take a break?
Yes,
Okay. We can take a like a 10-minute break and then we come back to specific activities. Any questions? No? Okay. So now I will give you a more specific knowledge of these capabilities. We start with herpes simplex or SHSV. There are two HSV1 and HSV2, two types of HSV. So basically, HSV1 is usually associated with cold sore, and usually you get this virus when you are a baby or when you are a child. In general, it affects anywhere above your chest. Okay, you get it from most of the time. You get it from your parents when you receive a case from them. HSV2 usually is associated with genital infections, and usually you get this after you become adult, become sexually active. And these two viruses actually are very similar in terms of structure. They are very close. They are very closely related, basically. They share a lot of homologies. I mean, they are almost identical. They're very, very close. In general, people say that there's a general thing that is HSV1 usually affects anything above your chest, and HSV2 affects anything under your chest. But this is not absolutely true. Actually, both viruses can go either way. Okay, HSV1 can also infect the genital area, and HSV2 can also infect your anywhere above the chest. And both are basically very highly infectious, highly contagious. So here's what happens. Okay, here's what happens during the herpes 1, herpes 2, herpes simplex infection. Okay. Usually, virus particles are in the saliva, or tears, or genital secretions. So basically, they are in this kind of things, and that's why they can be transmitted easily. They can transmit through contact. Okay. So contact, okay, basically, case or external contact, you get this very easily. So, as I already talking about earlier, when the herpes simplex virus first attack you, they will attack the mucosa, epithelial cells. Okay. So during this acute infection process, the cells, infected cells will produce new viruses, and the cells eventually get lysed, and that's why you have a lesion, right? Because the cell get infected, the cell produce virus, and the cell die, and in the same time your body, your immune system respond. So that's why the disease occurs first time, and most of the time it generates a lot of pain and inflammation. Okay, so this is acute infection. Okay, and this will result in the production of new viruses. Okay, the newly produced virus can travel through the sensory neurons. Okay, and get to the central nervous systems. Okay, for herpes simplex 1, eventually the virus will get into the trigeminal ganglion. Okay. And for herpes simplex 2, it will travel through the central neurons and get to the spinal cord, okay, ganglia. Well, the virus establish latency. Basically, during this time, the virus become quiescent and silent. There's no virus being produced. Okay. From time to time, as I mentioned, the latent virus can reactive, right? And can reactive and then goes back to cause the new infection. So this is just this picture, this slide just tell you the ganglia are located in the brain. For example, the trigeminal nerves, they are located in this area, okay? And the spinal cord is here, right? So for herpes simplex 1, they travel to the trigeminal nerves where it's established latency. For herpes simplex 2, they establish latency in the ganglia in the spinal cord, right? Again, so one, as I mentioned, during primary infection, acute infection, the virus attack the epithelial cell, right? Where the virus will reproduce and generate a lot of new virus, and usually from the virus will travel through the central nerve to get to the central nervous system, and where there is latency. And when the virus reactivate, so one thing which is very funny is the virus reactive the virus from the central nervous system, they will travel back to target to cause the new infection through the same central tract. Okay, so and then you will when the virus come back from the central nervous system to attack the epithelium, you will cause another recurrence for your epithelial infection. So that's why most of the time when you have a cold sore, for example, it will often occur to the same area. Every year when you get herpes cold sore, usually it happens to the same area, the effects are the same area. Because this is what happens. Because when the latent virus is reactive, when the really new the reactive virus will travel through the same tract and go back to infect the same area. So that's why occurrence usually occurs to the same place when you first get infected. So we'll give you some manifestations each of these herpes virus can cause. First of all, the first one is something called acute gingival stomatitis. This is something that can particularly occur to children actually. This is what happens when the herpes simplex attack the ganglia, the gingival. Okay, so and that's how it looks like. Okay, you see the gum becomes so red, and that's because you have inflammation, and the virus attack the mucosa, right? It can make it can give you a lot of pain. But this is generally this is a self-limiting disease. As far as you are immune competent, after a few weeks or even around 2 weeks, it will disappear. I mean you will recover basically. But it's if you are immune compromised, okay, immune suppressed, this can last much longer. It can be a big problem. So this is something that as a dentist you should be aware of. So when you see this, you should be able to tell your patient what they have. It's quite often symptom that you are going to see. In particular in HIV patient, because they don't have immune, their immune system is compromised. So you will see a lot of this in HIV patients.
Is there anything that you can do for it?
Yes, I will get there because we have anti-herpes virus drugs that is quite effective. Okay, so I will get there for about treatment. So this is how when the herpes virus attack you in your face, your lips, in the oral, your tongues, etc. So okay, so this is what happens, how it looks, and this is how it appears during the early time when you have a herpes simplex infection, and it's red, okay, and it's painful, and it can last several weeks, okay, and the symptoms for your lips, tongues, and the faces, your face as well. In general, it's pretty painful, and you have inflammation because your body is trying to get rid of this. So you have a lot of inflammation. You have a lot of immune cells, like NK killer cells. They come to get rid of this. Okay, get rid of the virus, clear the virus. At the same time, get rid of the infected cells, and that's why you have a lot of pain. So I mentioned, this is the early, this is how it looks like during the early time when you have a cold sore. But this time, this is how it looks like. Okay, eventually there will be disappear. They will disappear. So here is a because I want you guys to take a close look at this how the cold sore look like. And there's other concept called sore warts. Okay, so I want you guys to pay attention to this because there's a big difference between cold sore and warts. First of all, warts. Warts are caused by a different type of virus that I'm going to talk about tomorrow. It's caused by maybe papillomavirus or HPV. But in terms of the appearance, there's also a difference. First of all, cold sore, they can, as I mentioned, you are going to overcome this within like a couple of weeks, right? So first we see a red lesion, and then the lesion will become yellow and then it disappear after. But warts is kind of quite different, and the warts are caused by papillomavirus or HPV. And in terms of appearance, first of all, you don't see the red color because there's no inflammation. And secondly, the warts, once they are here, they will stay forever. If you don't treat them, the warts will stay there. And not only stay there, but also you will have more and more of this, and they will grow bigger and bigger. You will have more and more of these warts if you don't treat them, get treated. Secondly, usually these warts, you don't feel anything, even though they don't look like good, but they don't really bother you. I mean, you don't feel anything. It's not painful. Just be ugly, but that's because there's no inflammation. And I will talk about tomorrow. Okay. So, but you should be able to distinguish this because there's a clear difference between cold sore and warts. Cold sore, basically you can see they will develop in progress from the red color to yellow and then disappear. But warts, the wall will stay there and get bigger and bigger, more and more.
And how long did you say it takes for it to progress from the red to the yellow?
Usually it's within 2 weeks. Yeah, usually get recovered within 3 weeks for sure. As far as you will competent, you will overcome this. And of course, the herpes simplex can also affect your eyes, particularly in the kids, and also the brain. And this for little kids, it's very dangerous actually. The virus can get into the brain and cause meningitis, as a fetal. And so, if you see little kids get infected by herpes simplex, you need to know that you need to get treated immediately because it's quite dangerous. Particularly those newborns, because their immune system are very weak. Any question? Okay. This is how the genital herpes look like. Infections when you have a genital infection, that's how it look like. And actually, in the US, genital herpes is very, very frequent. So you guys need to be aware of that. So in terms of diagnosis, the traditional, the conventional diagnosis is based on microscopic detection. Basically, when you scratch the lesions, you look at the, what you get under the microscope, you see big or giant cells, and also you see these kind of secretions, these particles, that come from the cells. So these are typical. This is how they look typically under the microscope. But right now, we don't do this anymore because there are so many, diagnosis tools available, molecular or immunological diagnosis, tools, technology available. We can make a diagnosis within seconds, minutes, you see. So now I get to your question, treatment. Okay, for herpes simplex 1 and herpes simplex 2, so far we don't have a vaccine that is very effective. People have tried, very hard to develop a vaccine against herpes simplex, but so far, as a failure, we haven't been able to do that. But the good news about herpes is we have very effective drugs that are available to specific target herpes virus. Okay, and these are the names of these drugs, but they look very similar in terms of structure. I will show you here how these drugs work. This is something important because you need to understand under herpes virus the mechanism how they target the virus. Okay. As you know, because this is when I come back to your biochemistry. Okay. So as you know, the DNA or RNA molecules, their basic structure is a nucleoside, right? So in the nucleoside, you have 5 carbon sugar and also a base, right? So in the sugar group, you have 2 groups, one OH group here and one OH group here. Okay. In this OH group, during the synthesize, this OH group will make a triphosphate bond, right? And so during DNA synthesis, okay, this triphosphate group will make a bond with the next nucleoside with the OH group of the next nucleoside to make a bond. So that's why the DNA chain can elongate and right? That's how the DNA synthesis continues, right? So what happened with this anti-herpes virus drugs is that we made nucleoside analogs. So in these analogs, we replace this sugar with something like this. Okay. So in these molecules, we have only one group, one OH group. Okay. And this OH group can be phosphorated, basically because they are triphosphate bond. Okay. And this requires the enzyme that comes from the virus. It's called herpes virus encoded, herpes virus specific thymidine kinase. Okay. So this virus encoded thymidine kinase will add the first phosphate to this group, and then the cellular phosphate thymidine kinase will add the second and the third phosphate to this group and to make a triphosphate. Okay. This is very unique because the host and the cellular thymidine kinase cannot do that because this is something that is foreign to our body. Right? Our cellular thymidine kinase do not have this ability to triphosphate these molecules because our system, our kinase, thymidine kinase is only can do that to the nucleoside. Okay. So the virus encoded thymidine has such a unique feature, that can add the first triphosphate to these drugs. Okay, that's how when you add this drug or when the patient takes this drug, okay, these molecules will be incorporated into the DNA that's undergoing synthesized. Okay, and what happens? It will be added to the nucleoside and upstream nucleoside of the DNA, but will not allow the addition of the next nucleoside to the DNA chain because the next meets the other OH group. Okay, that can make a bond between the triphosphate and the OH group of the next nucleoside. So basically, this is how it disrupts the DNA synthesis. Right? So now, I have a question to you now. So, do you think this drug can also block the DNA synthesis of the host cell that is infected by this virus? What do you think?
The cell uptakes the medicine.
But isn't it herpes very specific?
So if the cells are infected by this virus, right? If the drug can inhibit the synthesis of the virus, can this drug can also block the synthesis of the host cell?
Oh, you're saying so cells that are not infected?
No, I'm talking about the cells that are infected by this virus. So in the cells that are infected by this virus, the drug can block the DNA synthesis of the virus, right? My question to you is, can this drug can also block the DNA synthesis of the host cell?
The host cell doesn't use that kinase, right?
Yeah, but this host cells, when the cell is infected by this virus, this 17 kinase virus, including the 17 kinase, is present in the cell. So, what do you think?
Yes.
It definitely will block the DNA synthesis of the infected cells because the enzyme is present in the cell. So basically, in other words, the drug can not only block the virus, but also kill the cells because the presence of this 17 kinase.
So as long as someone's taking one of these medications,
Yes.
are they able to pass it on to other people, or what exactly does, if it doesn't cure the disease, what is it doing for the patient as far as transmission to someone else, or?
So basically, will stop the virus replication. So if the virus replication is stopped, you cannot produce virus. So if you don't produce virus, obviously you cannot pass the virus to somebody else. Got it? All right. So here comes the other questions. So if the drug can target both the virus DNA and also the cells that are infected by the virus, so my next question is, can this drug also block DNA synthesis of the uninfected cells?
No,
Why?
because they don't have the thymine antagonist.
Exactly. And so that's why these drugs, they are so specific. They have very little side effect, and they are so selective. They don't have much effect on the uninfected cells, the other type of infected cells. Right? That's why they are so good and so specific, so selective. Now, here comes the other question: can this drug also get rid of the latent infected cell, the virus? Why? What's the reason?
It would only affect actively dividing cells, right?
And what's the reason?
Those are the only ones that are making more DNA.
Think about it. What happens during latent infection, and what happens during acute infection? So what happens during acute infection? The virus genes are expressed. So you have the expression of this 17 kinase, right? And during latent infection, what happens? During latent infection, the virus gene becomes silent, and you don't have any virus gene expressed. So in other words, you don't have this 17 kinase, right? So that's why the drug does not have any effect on the latent virus. So even you take a drugs, not the virus drugs, the virus still is in your body. You can never get rid of it. So far, we don't have any drug, any treatment that can remove the latent infection.
So the patient just has some baseline serum amount of this drug in their system at all times,
Yes,
ready for the moment that it comes out of its latent phase.
yes. So the drug can keep the virus in check, basically,
Okay.
but you can never get rid of the virus completely. Because as far as you stop the drug, and as far as your immune goes down, and the virus reactivate, it will come back. So that's what happens. Any other questions? Do you have any questions?
Can you go back, you were explaining about the OH group and the triphosphate bond, I didn't really understand that part.
Okay, so,
Because don't you still have the OH group on the end here?
So when you doing DNA synthesis, right? You need what happens is the OH and the the how do I let me rephrase. So the what happens during DNA synthesis is that the triphosphate group of your next nucleoside is going to make a bond with the OH group here of the upstream nucleoside. So it makes a bond. That's how the DNA chain progress, right? So if you don't have this, if this molecule is incorporated into the DNA chain, so it will, but that's basically it will be added to the chain because it will form a bond between the triphosphate and the upstream nucleoside, the OH group. Okay, so it will be added, but it will not allow the next nucleoside to be added because the next the OH group that can.
Okay, the upstream.
You got it right? Okay, that's how it works. So the other thing I want to mention about this herpes drugs is they are basically effective for all types of herpes virus. And all types of herpes virus, even though they have some difference in terms of structure, and the sequences, but their function is very similar. They all have this herpes virus included 70 kinase that can metabolize, make this triphosphate, to the drug. And our host 70 kinase cannot do that because we are genetically made that way, And the herpes virus, including 70 kinase they can do that. So because of this, this feature of this herpes virus, 70 kinase, people trying to take advantage of that. So what we do is we use this feature to actually to kill cancer cells. So basically what we do is, so there are people who make recombinant virus that can express this virus, including the 70 kinase, under the control of a promoter that is come from tumor cell. So in other words, once you infect the cells with the recombinant virus, the tumor cells will produce and express this 70 kinase. This virus including 70 kinase. So if you treat the patient with the cancer patient with these drugs, only the cancer cells that express this kinase will take these drugs and get killed. So it's something that is very useful and for cancer treatment. But this is really relevant to this lecture. I'm just talking about this feature of this herpes virus enzyme.
In that case that you're talking about, how would you prevent the healthy cells from taking up the virus?
You mean talk about the treatment of cancer?
For example.
Yeah. So that is why we develop something called uncolytic virus. So basically, we use the other virus to express this virus protein, virus genes. Because in the recombinant virus, we also put a promoter. This promoter comes from a tumor cell. Basically, this promoter only is active in tumor cell. In normal cells, this virus candidates cannot be expressed. So only tumor cells get expressed. Okay, so that's why the drug is specific for tumor cells, not cancer cells. Now I'm going to get to another alpha herpes virus, which is the varicella zoster virus, which is very common. I believe everybody probably in this room get this virus sometime, at some point of your life. This is a virus that can cause two diseases, okay? But it's the same virus, but it can cause two diseases. One is called, varicella or chickenpox. I don't know if you guys have already gotten that. You're near the baby. How many of you have chickenpox before? Oh yeah, I can see. Yeah, most people get this, when you are, child. And I think right now, because of the vaccine, the baby, newborns, they get vaccine probably 3 months after birth. So this disease is getting less and less frequent. But still, a lot of people, they don't get it, they don't get a vaccine, so they still get, chickenpox. And as I mentioned, this is an alpha herpes virus, and once you get infected, and once you recover from the chickenpox, eventually the virus will travel to your brain and have a little infection. However, when you become, when you become old, your age enter the middle age, for example, 40, 50 years old, when your immune system, start to going down, the virus can come back again and strike you again. So basically, the little virus reactivate, and then cause infection, and that is called shingles or herpes zoster, or shingles. Usually occurs in adult, and basically, this is another disease that's caused by the same virus, because basically it's a reactivation. Okay, this shingles is caused by is because of the reactivation of the same virus. So this is how you get chickenpox. Basically, when you first get the virus, usually through contact when you receive kiss from your parents, you get the virus, and once the virus enters your body, it will go through the respiratory tract and spread to the lymph node, and it takes actually 2 weeks for the virus to get to your skin, okay, and will it infect your epithelium, your skin cells. So basically, that's when you see chickenpox, those skin rashes. Okay. Usually, the skin rash, when you get this kind of chickenpox, it's all over your body, it's everywhere. It's kind of skin lesions. And usually, and again, this, this is a self-limiting disease. Usually, you don't need to. For children, you need to get treated. But if you are if you over 10 years old, even without treatment, the virus eventually will be cleared by your immune system. And so self-limiting disease basically. And eventually, the virus will travel to your ganglia, will establish latency. Okay. So as I mentioned, the virus can reactivate, okay, and then make a new virus, and then it travels down through the same sensory nerves to infect your skin again, and that is when the shingles occurs. So there's a big difference between shingles and chickenpox. So when you have shingles, usually it affects one side of your body. It's not like a chickenpox. Chickenpox usually when you have chickenpox, it's like everywhere on your body. But when you have shingles, usually what you see is it affects one side of your body. So the question to you is why this happens? Why shingles occur only at one side of your body? So this has something to do with the structure of your nervous system. So usually you have two hemispheres in your brain, right? So your right hemisphere, the nerves are connected to your left side connected to the right side of your body, right? And the right hemisphere of your brain are connected, to your left side. Okay. So basically, the reactivation really occurs locally, somewhere in your brain, right? So when the virus on your left side or on your right side reactivate, you will traverse through the sensory nerve to the other side of your body. And that's why you have effects on the one side of your body. Okay. That's a difference between shingles and chickenpox. And the shingles not only can affect your skin, but also can affect your face, your eyes. And when it attracts your eyes, it's very dangerous because it can cause blindness. Some people, they get, when they get like 40, 50 years old, and then suddenly become blind, and they don't have any idea why it happens. And actually, many of the times it's because of this herpes virus. Okay.
Wouldn't you only become blind in one eye?
Yeah, usually it affects one eye. Yeah, that's right. So in terms of diagnosis, for this virus, it's very obvious because when you get a chickenpox, you get those skin rashes everywhere, and usually it occurs to the kids, so it's very easy to diagnose. And chickenpox is the same thing because when you have chickenpox, you also see the kind of rashes, and also when you have pain, sharp pain. So it's quite easy to make a diagnosis but of course we also have other techniques to make a more accurate, diagnosis. If you go to see our doctor, you will take a blood, draw blood, and make a diagnosis based on based on molecules, DNA, RNA, proteins. They have all different kinds of ways to make a accurate diagnosis, basically. But in terms of prevention and treatment, chickenpox visibly is unique because this is the only virus so far we have a very effective vaccine available. So for other herpes virus, we don't have a vaccine so far. But for visibly, we do have a very effective vaccine available. I think everybody probably already received this vaccine. This vaccine basically is developed by a Japanese scientist. Basically, it's a lab. So this doctor tried to culture the initial, the original virus in the laboratory, and during that process, for very long term, many years, during that process, the virus somehow mutated, become less pathogenic. We call it attenuated live virus. Okay, so this virus can be produced in a laboratory. Okay, in the culture, cell culture, but the virus still can infect, but not cause any disease. So that's why we use it as a vaccine. In terms of treatment, if you do get shingles or your kids get chickenpox, there's drugs available. It's the same drug that I just described you. And also in the same time, I'm pretty sure your patient, your doctors also will give you other drugs like anti-pain, anti-inflammatory drugs, okay, anti-inflammation drugs. So because when you have shingles or chickenpox, you have pain, sharp pain, very, very painful. And at the same time, you have the inflammation. So if you don't control, it will cause some other conditions. Do you have a question?
Yeah, does the vaccine protect against both chickenpox and shingles?
No. Oh yeah, that's a good question. That's a good question. I think right now there's a vaccine available for shingles as well.
Okay, so you have to get 2 separate vaccines?
Yeah, it has to be 2 separate. I don't know why, but this is what happens. I don't have an answer for you on the why the same vaccine does not prevent shingles. That's something I don't know actually.
But how is it possible that the chickenpox, so if you have a chickenpox vaccine and you never get chickenpox, then can't you never get shingles because you need the latent virus? Or can you get shingles without having chickenpox?
I think for shingles, you have to get chickenpox first.
Right.
If you never get chickenpox, it's very rare for you to get shingles.
So if you use the chickenpox vaccine and you never get chickenpox, then you would not need a shingles vaccine.
That's something I'm not sure.
I think the shingles vaccine exists primarily because you have a lot of baby boomers who did get chickenpox, but now potentially could have a reactivation. But I think that what she says about the chickenpox vaccine, if you've never had chickenpox, then yeah, you wouldn't get the subsequent. But, you know, the shingles vaccine, I think, is for people who did have chickenpox before the vaccine was made. What was that, 1995 or something?
Yes, that's something I don't think we have a very clear answer yet, to be honest. But I do know that there's a vaccine that is coming for shingles, specifically for shingles. Maybe the
Oh yeah, they sell it but you can get it at Walgreens, CVS,
yes,
whatever.
yeah. But I don't know how different it is than the shingles, than the initial chickenpox vaccine. I don't know whether there's any difference between the two or not. That's something I'm not very sure. I can I can do a search and tell you tomorrow. Okay. Let's see how I do this. Yeah, I think probably. Do you guys have any other questions? So if you guys do not have any questions, probably we will stop here today, and we'll continue tomorrow. How about that?
Thank you.
Yeah. But if you guys have any suggestions to me about this lecture, whether I make it clear or not, let me know. Okay.
Thank you.
Always think it's your best.